Hi, everyone. My name is Jonathan or Jono Abshier out of Portland State, specifically in the Extreme Virus Lab. And I'm going to kind of throw a wrench in things and talk a little bit about viruses and the extreme ones that I study. Don't really need to go over the title of my talk, but our journey really begins here. This is a location in Northern California at Mount Lassen, where several of these volcanic hot springs exist. And in addition to these hot springs, we have these guys inhabiting them, what I refer to as microbes from hell. Over here on the right, we have a boiling mud pot. So these are volcanic hot springs, and we typically study these because we understand them to be analogs for several exoplanets, inside and outside of our solar system. Again, this one is in Lassen National Volcanic Park. And one of these such organisms that we find in these locations is Saccharolobus solfataricus species. So it is a hyperthermoacidophile that really likes these hot and acidic conditions that these volcanic hot springs make around 80 degrees Celsius and really low pHs at 3. Over here on the left is a scanning electron microscope image of the Saccharolobus that we isolated from this particular location a few years back. And when we think about these environments and these cells existing, I often come to the point of the most abundant biological entity on the planet, which is viruses. And alongside these S441 organisms, we have a spindle-shaped virus, a really unique virus that I work on, Sulfolobus spindle-shaped virus 1, as you can see in this transmission electron micrograph. It has these unique spindle shapes that interact with the cells. Here you can see an archaea, so an archaeal flagella running through the micrograph. It does this really interesting thing structural-wise where it makes these rosettes, and we think that's some sort of temperature stability kind of adaptation. And when we think about these things and how they interact, we go straight to host interactions and just survivability in these environments. And one such system that we think about are toxin-antitoxin systems and how things are interacting. These are also known as addiction modules, first found on plasmids, really characterized within prokaryotic genomes today. Tons of bacterial addiction modules are already characterized. There's a few different types, but typically they look like two genes that are transcribed right next to each other and kind of confer this mechanism known as post-segregational killing, which we can see here on the left. We have a plasmid that utilizes a TA system, so has that toxin-antitoxin phenotype, where you have a mother may produce clonal, similarly offspring. One of the offspring might inherit this plasmid, and so long as it keeps that plasmid in these particular conditions, you confer normal growth, whereas if you lose the plasmid, you use that ability to mitigate the toxin-antitoxin system and result in death, where in a system that's negative for that, you still see normal growth overall. And kind of characterized in a community setting, we view it like this in a series of uncolonized cells where an event might happen, in particular, in this case, a virus infection, a viral infection. You have this addicted survivor that has that genome and that ability to mitigate the effects of the toxin, where you have culling of these other cells that just don't have a way to do so, which then goes on to replicate and confers this sort of protection among this population, where there might be another group encounter later on, and they may outcompete that particular group, resulting in more culling. A majority of my work, again, has been on the spindle-shaped virus, or SSV1. Largely, a ton of mutagenesis studies have been done by previous graduate students before myself. And what they did essentially was throw insertions into several random parts of the genome just once, and viewed really how phenotypically different and plastic the genome was. I focus on two open reading frames in particular that I believe to be the toxin-antitoxin system. Over here, this TX and T3 open reading frame. So we know largely that knocking out or changing up the antitoxin still results in infectious virions, as we will discuss in just a second, as well as deletions and insertions for that particular open reading frame. But T3 seems to be crucial, which in theory would be the toxin, the thing that's having the activity on the cells. And to talk a little bit more about virus reproduction, for those of you who aren't really familiar with virology, viruses can do really two kinds of life cycle or a combination of both. Here we have the lytic cycle in which the phage DNA, goes into the host. Eventually it's replicated, assembly starts to happen, and that cell will burst. We typically look at this through plaque assays, which is just kind of plating a lot of cells, allowing them to grow and observing clearings where this burst is potentially happening. Another reproduction or life cycle that viruses can do is a lysogenic cycle, in which they have their DNA inserted into the host, or RNA, will kind of incorporate that into the chromosomal genome of the host, in which you still get replication and future generations. Some sort of induction or stressor event might happen where they switch to the lytic cycle. Eventually there's that assembly and then bursting. Again, we visualize that through the plaque assay. SSV1 has a very specific phenotype in that it doesn't actually burst the cell when it leaves, it buds from the cell. So pretty unharmed in terms of what the cell looks like. And we test this in the lab through a version of the plaque assay known as a halo assay, in which we still grow a lot of cells and we spot various concentrations of viral supernatant and observe these clearings, although we know that this burst step isn't happening, and at least it seems to be the toxin as the causative agent. So looking through this toxin, I decided to do some genetic analysis, looking for specific sites on the toxin. This is its structure right here. Again, that A291 open reading frame. This is an alpha-fold predicted structure. And according to signal P, it does look like it's a signal peptide with a cleavage site at position 25 and 26. So what I sought to do initially in my project was look at some of these mutants that were already available to us using halo assay and screening for virus replication. Over here, we have a screening for replication, which I'll get into in a second, but we have our halo assay over here on the right. And largely within our wild type populations, we consistently see that clearing. So we understand there to be toxin activity. This is just across two passages, these same mutants. This is another wild type that we actually have an insertion. It doesn't do much to that particular genome. It seems to be not affecting infectivity. This 254, 291, those are two insertions that are directly into the toxin open reading frame, and they seem to knock out function. And then I chose one mutant to substitute by just switching the residues at that particular site to see if I would still get activity or cleavage at that particular site. And you can see that among our wild type populations, we get clearings. In our insertion mutants and our substitution mutants, we don't actually see any clearings. But when we screen for virus replication, we do continue to see at least levels of virus replication. So we know there are active virion particles, and we did go on to at least visualize these on the TEM, the electron microscope, to make sure that there were viruses in these particular supernatants. So we do see at least active replication and budding of viruses from cells. And in more of these genetic analyses that I've done, we have seen at least some of these DALI searches that come back when they align structures to others. We do see them encoded in host genomes of other thermophiles, other hyperthermophilic, other Sulfolobus species. The ones indicated with the virion are actually virally encoded proteins that have very similar structures. This is something that I'm at least hoping to follow up on. And then kind of bringing it all back, we think about, looking in hot springs on Mars. We think about maybe these hyperthermophiles and their viruses, any biosignatures. But ultimately, this toxin-antitoxin system seems to be allowing for persistence of these particular populations in these environments. And we see that through evidence of horizontal gene transfer and just persistence in these stressful environments while being actively virally infected, allowing for some phage protection in some cases. That's still being studied. Just sharing a little bit of a sampling video with you from our one of our most recent trips. We just go into the hot spring with a conical tube, bring it home, enrich it, and see what we can pull out of it. In this particular area, we isolated the host organism. A few yards away, we actually isolated another spindle-shaped virus 10. Not completely infected. We're still looking into the host organism that we isolated and actually pending publish of that genome. But why study these archaeoviruses and why hunt for these viruses? So we can look at it in the context of comparative virology, looking at what viruses are existing today, affecting human health and their role in pathogenesis. We can look at these host virus interactions in these environments and how they do confer that persistence and allow the organism to thrive while still being infected. Again, the role in these ecosystems, what role the virus has on the host organism in terms of the environment and other populations within these really diverse and extreme conditions. And then just evolutionary history. Ultimately, it's understood that viruses, maybe predominantly came before the cell. In an RNA world, there's a lot of info on that. And I just think it's a really interesting concept that evolutionary would make sense. Especially on other exoplanets. And then, yeah, just also looking for more of these organisms on a yearly find. We can isolate these organisms and study them, characterize them if they haven't been characterized, and see how they might interact with a virus where they haven't been before. And with that, I just want to thank the Extreme Virus Lab, some of our funders, and you guys for coming today.